Hello, dear students. We have already started with our new chapter, that is a biological classification of class 11th. In previous lecture, we have already completed the classification system, kingdom Monera, kingdom Protista, kingdom Fungi. And today, in this lecture, after the completion of fungi, we will be dealing with the kingdom Plantae, kingdom Animalia, some general information regarding this. Then we will talk about the viruses, viroids, prions, lichens, and mycorrhiza. So, in today's lecture, I welcome you all on behalf of Sri Jayendra Rangjone classes. Okay, dear students, so let's start with the kingdom Plantae, general information. And in general information of kingdom Plantae, we will discuss that you know that kingdom Plantae, when we hear this term, in our mind we can relate that this is going to talk about the plants. Now, you also know that plants are the photosynthetic organism. They can synthesize their own food and they are autotrophs. And for this, they require pigments, and that pigments are present in the membranes of a chloroplast, okay, or within a chloroplast. So, if you talk about kingdom Plantae, this kingdom Plantae includes all eukaryotic chlorophyll-containing organisms, commonly called plants. So, it includes what? All eukaryotic chlorophyll containing organism, and this chlorophyll is a pigment which is found in case of chloroplast, and due to presence of this, you can see these are the plants, you know very well, they can synthesize their own food. So in this, we will also talk about a few members who are partially heterotrophic. They are not completely heterotrophic, they are partially heterotrophic, such as some of the insectivorous plants or parasites that are also included in clinical plant. Then, when we talk about the examples of the plant, there are some insectivorous plant examples are like a bladderwort, Venus flytrap. These all other particularly we can say a insectivorous plant. You can see this is the example of bladderwort and this is the example of Venus flytrap. These all are insectivorous plants. So by using the insects, they get nutrition. This is Cuscuta is also a complete parasite. It is a complete parasite which is completely dependent on another plant for the nutrition, that is for water as well as mineral and food. Then, when we see the plant cell, we see that plant cell have a eukaryotic structure with a prominent nucleus. Actually, plant cell have what? Eukaryotic structure, the prominent nucleus, prominent chloroplast. Now, the structure of chloroplast is this, and in this structure of chloroplast, you know that when we see the structure of chloroplast, we come to know that there is an outer membrane, there is an inner membrane, means chloroplast and double membrane form. Then, when we see internally, this particular whole stack-like structure are there, that is known as thylakoid. But there are multiple thylakoid are stacked over one another, like this. So, like coins, a stack of coins, thylakoids are nice. These thylakoids are connected by a structure that is known as stoma lamella, and the fluid which is found here, that is stoma. The one phylum of this thylakoid is known as granum. Granum is a stack or phylum of thylakoid, and the space which is found between outer and inner membrane, that is known as intermembrane space. So from here, it is clear that chloroplasts are double membrane bound organisms. Then, when we talk about the cell wall of these plants, cell wall of plants are made up of cellulose, means they have a cellulosic cell wall. When we talk about the plant, they also synthesize the food, and after synthesis, they also store the food, and that food is stored in the form of starch. And plant also show a absorptive mode of nutrition, means they absorb nutrition from the surrounding areas. When we talk about plant take, the plant take includes the algae, first the organisms which are going to be included in the plant take, that is algae. And you can see this is the algae blooms or algae which are growing in the seawater, which is shown in this diagram. Then the another group is bryophytes. These are the bryophytes which grow in some of the shady places. And these are pteridophytes. These are also found to grow in aquatic places. Then gymnosperms are known to study. That are the seeded plant in which we talk about the pinus and cycas. Then there is an angiosperm. And angiosperm are the flowering plants. These all are included in the plant. And after the completion of the biological classification chapter, the next chapter we are going to discuss the kingdom plant. Which kingdom we are going to study? We are going to study kingdom plant. After kingdom plant day, what plant day includes that we will discuss. How the perform nutrition that we will discuss. That is the general information according to the NCERT. But detailing of algae, bryophyte, pteridophyte, use all this we are going to discuss the next. We have to go through some initial information of kingdom Animalia as well. And we talk about when kingdom Animalia includes animals. So the kingdom is characterized by heterotrophic eukaryotic organisms. Yes, animals cannot synthesize their food, so they are heterotrophic, they are dependent on another organism. But with that, they are eukaryotic as well because they have a eukaryotic structural organization where they have a membrane bound organelle, where they have a nucleus that all are found in their cells. So they are known as heterotrophic eukaryotic organisms. And these all are the animals which you know very well. There is no need to explain animals, what they are. That are multicellular and their cell lake a cell. One of the important features of the animal cell is that the animal cell was they are devoid of cell wall. And if they are devoid of cell wall, the outermost covering in their cell become plasma membrane. Now, when we talk about animals are directly or indirectly dependent on plant for food. Directly, they are herbivores. They are going to eat directly what of plants and they get their food. But if they are carnivores, they are going to feed on the animals like herbivores. But herbivores also depend on plants. So indirectly, these carnivores are also dependent on what food, so indirectly also on the particular plants. When we talk about animals intake their food and after the ingestion of food, they digest this food in their internal cavity, the digestive tract, the digestive system. And after this, they absorb the food and they particularly store that food as a glycogen compound. So, if somebody asks, what is the reserve food material in case of the plant animalia or in case of animals, then there is nothing but a glycogen and fat. So, this is the reserve food material of animals. Okay. Then if you talk about their mode of nutrition is holozoic, that is ingestion of food by mouth, they ingest the food. Then different activities occur. Apart from this, if we say the food organisms are there, invaginations are there, and food will get seen. This is shown in the cell. This is shown here by a cell. Suppose this is the cell, and this is the particularly food organism. So the invagination develop around this food organism. And then the food is taken inside the body that we enter the food into. Okay, so this is how they take food. They take food that is ingestion by the invagination of plasma membrane, and then this food is going to present in the form of food vacuole. So when we talk about animals, they form a definite growth pattern and grow into adult, have a definite shape and size. And in that, we can say the animals follow a definite growth pattern if they have a definite shape and size. and they grow up to a certain age of an individual. They cannot grow throughout the lifespan. Whereas in case of plants, we can see that the plants grow throughout the lifespan. If we cut any section of plant, any stem of plant, it will regrow. But in animals, if we cut a head, it cannot regrow. So because of this, animals can be showing in the definite growth. And in definite growth, also different stages are there. And in that stage also, you know that there is a preterm uniform infant, then there is a 0 to 28 days newborn infant, then infant to toddlers, and then 23 months, then there is children from 2 to 11 years of age, and then from 12 to 18 years they reach to the adolescent stage. So the higher forms show a elaborate sensory and neuromotor mechanism. Okay, so when we talk about higher forms of animals, they have a elaborate, detailed sensory and neuromotor mechanism. Apart from this, most of them are capable of locomotion. Yes, most of them are capable of locomotion. And in that, the sexual reproduction is by copulation of male and female, followed by embryological development. They locomote, they have locomotory organ. And when we talk about sexual reproduction in animals, the male and gamete fuse, and after fusion, a zygote will form, and zygote will develop into the embryo. So embryo is also found present in case of animals. You can see this is the particular different types of animals and how they are locomoting that can be observed. Let's talk about some of the question. Cuscuta is a particular plant which is known as complete parasite, as we have discussed, or we have seen in the diagram. They are dependent on another organism or water, mineral, as well as food. So they are known as what? Complete parasite. Then when we talk about eukaryotic chlorophyll containing organism, commonly called as eukaryotic chlorophyll containing organism, means they are nothing but plants, and these plants are placed in kingdom planting. So the answer becomes option 2. The third question is cell wall of plant is made up of. Yes, we have discussed the plant have a cellulosic cell wall. So cellulose becomes the correct answer. And after this kingdom planted kingdom animalia, we will be going to talk about viruses, viruses, prions, and lichens. Okay. And this is our major focus. That will be major focus on this component in this section. Dear student, these viruses, viruses, prions, and lichens are not classified by RH. They are not classified by RH by kingdom classification. So, if you talk about them, they are not classified. So, in the 5 kingdom classification of Botany, it is known that the lichen and some acellular organisms like viruses, viroid, and prions are not included in 5 kingdom classification. Then, when we talk about viruses, you can see this is the diagram of virus, that is bacteriophage. We will discuss the structure in sheet, but one thing you should locate that in this virus there is a presence of protein coat and there is a presence of sheet and there is a presence of DNA. Two things you should remember: the presence of DNA and the protein coat. So this organism did not find a place in classification. Viruses have not found a place in classification. And since they are not truly living, they are not truly living, because of this they are not classified in this classification. Then they are living as those organisms that have a cell structure. First, they are not classified because they are not truly living, but they are considered as living as those organisms that have cell structure. Why? We will discuss that. How? So now we will be talking about first viruses in detail, then we will talk about other subviral agents. So let's talk about viruses in detail. So the term virus, when we discuss the question first that arise, either they are living or not. what is the living state? So we have discussed already in the living world chapter that those satisfy the defining properties, okay, that will be considered as a living. So the state of being alive is living state. Then what is non-living state? Non-living state, the thing which not endowed with life, means which does not have a life, which does not have a defining properties like consciousness, increasing growth, metabolism, they will be considered as a non-living state. But this is also showing a living thing and non-living thing. That living thing are the protist, plants, animals, fungi, bacteria, all are living things. But non-living things like water, soil, air, light, and this all are non-living things. But when we talk about virus, this virus are particularly neither living nor non-living. Now the question is why life depends on physical and chemical process that you know, that is metabolism specifically we talk, and like consciousness. This all is going to define the living state. So when we talk about the smallest organism called a microorganism arises from the less number of cells. Yes. And when you see this diagram, this is a particular pictorial representation of the virus that we meant. So when we talk about further, come to know that viruses are unique group of biological entities. They are a unique group of biological entities. They are known to infect every type of cell. Either it can be a plant cell, animal cell, or it can be particularly bacterial cell. It can be infected by viruses. So viral infection includes different organisms like bacteria, algae, fungi, protozoa, plant, animal. They can infect all of them. But it is known that viruses are host specific. Viruses are host specific. Means the particular type of virus is going to infect a particular host only. So this host specificity is one of the important characters. Why? Because if we talk about plant virus is going to infect plants only. Same way animal virus will infect animal only. Bacterial virus will infect bacteria only. Such a way they are host specific, so they show host specific. Then viruses are not composed of cells and that cannot be seen under a light microscope. Right? Viruses are not composed of cells. Okay, and cannot see under a light microscope because they are too small. And one more thing, they have not have a true cell structure. So viruses are nothing but particles. And this virus particles, single type of nucleic acid, either DNA or RNA. Dear student, this is one of the important statements that you should know. That in case of viruses, there is either a presence of DNA or presence of RNA. Both cannot be present. So here, which term we should mark? We have to focus here on a or word. Whereas in bacteria, that is mycoplasma, we have discussed that mycoplasma has DNA and RNA both. So, but viruses have DNA or RNA. Then, this is the structural DNA double helical structure which is found in case of viruses. This is RNA, which is a single stranded sequence. RNA is found usually single stranded, but DNA is found in double stranded. Okay, so this is the thing which is found in case of particularly viruses. Then, if we go on talking further, we come to know that in viruses, the nucleic acid are enclosed in protein code. In viruses, what is found? Nucleic acid is found. Either it is DNA or RNA, but this DNA or RNA are enclosed within a protein coat. And this protein coat is made up of specifically protein. And outside a protein coat, there is a presence of envelope which is made up of liquid protein and carbohydrate. Dear student, one of the important statements is because that this protein coat, if we talk, so in this, the nucleic acid are enclosed in a protein coat, and this protein coat is specifically made up of protein only. But outside protein coat, there is a resistant envelope. That envelope is made up of lipids, protein, and carbohydrate. So these all are combinedly form an envelope in some of the animal viruses outside the protein coat. So if we talk further, we come to know that viruses do not exhibit most of the life process of a cell. Do not exhibit, they do not have the different life process of the cell because they are innate. But they maintain their genetic continuity through multiplication and they also go under the mutation. They are certainly more than a lifeless molecule. They are just molecules. They are just molecule, chemical molecule made of protein and nucleic acid usually. Okay. Now the viruses are best described as infectious particles. Yes, they are infectious particles, but they are considered as neither living nor non-living, and they are made up of protein and nucleic acid. The study of viruses is known as virology. So it is a branch of science which deals with the study of virus. They are most specific, as we have discussed, the specific called or obligate intracellular parasite. They are most specific, they are most are specific. Second is they are obligate parasite. And they are obligate intracellular parasite means they cause a parasite. They use a particular host cell by entering within that host cell. So they are considered as obligate intracellular parasite. Then, if we talk further, we come to know that let's talk some of the questions of the sequence that we will discuss about this further. The study of viruses is known as, just we have discussed, it is known as virology. If it is of bacteria, it is bacteriology. If it is a science dealing with the technological field, that is the biotechnology. And if it deals with the microbes, that is the microbiology. Here, the study of viruses is virology. This virus are known as obligate parasites. So, which of the following are obligate parasites? Viruses are obligate parasites. Yes. Then what are best described as infectious particles? So obviously viruses are best described as infectious particles. And after this we are going to talk about discovery, classification, and structure of viruses. We will talk about discovery, classification, and structure of viruses. Till now we have discussed just general information of viruses. Okay? So I hope you are clear that either viruses are living or non-living, either they are particularly okay, so that we are going to discuss. So before studying this all parts, we will talk about some of the important characteristic feature of viruses which we have learned till now. Okay. And if we note down the characteristic feature of this, then first is the characteristic features of this particularly viruses, and that first one is are submicroscopic and non-cellular. Apart from this, if we talk, they are generally smaller. We have discussed also that they are obligate parasite third thing that we should find out that they are either DNA or RNA. Then it is known that there is not any virus or single virus which is both DNA and RNA. They are inert outside the specific host in crystalline form. Fifth one is they contain nucleic acid, so they are capable of protein synthesis with the help of ribosome of the cell. Apart from this, these viruses are also exceptional cell theory. So these are the particularly important characters with respect to. Now after this, we are going to talk about some of the particular discovery part. In discovery, we will discuss that the initially in 1892, one Russian pathologist was there is DJ Ivanovsky, that is Dmitry Ivanovsky, who was working on a tobacco mosaic disease. Okay, and what he did, he separated the particularly, we can say, microorganism from the sap of infected plant. Okay, the particularly plant of tobacco infected with the tobacco mosaic virus, they isolated a microorganism from the sap of this infected plant and named them tobacco mosaic virus. He also reported that these viruses are smaller than bacteria and they can pass through bacteria filters. So that was given by James in 1892. Okay, so the infectious agent passes through the pores of the bacterial filters, passes through the bacterial filter. The filters have all infected to healthy plant. The symptom of mosaic disease were seen in it. But after this, M.J. Bezernik was another scientist, you can see here, and the full name is Martinus Bezernik. He concluded that disease causing agent was a contagious liquid fluid. And contagious liquid fluid or infectious living fluid, that is nothing but contagium biome fluid, which says that Bezernik demonstrated that the extract of the infected parts of tobacco could cause infection in the healthy plant. As such, the fluid or is infectious, the living fluid is infectious, so the term is used that is contagion by fluid. So if somebody asks, you should note down that contagion by fluid is given by M.W. Bezernik. After this, another scientist was there whose name is W.M. Staley. W.M. Staley in 1935 he crystallized TMV, that is tobacco-mosaic virus, and he showed that these viruses are mainly made up of protein, and for this, Staley got Nobel Prize for the discovery of this crystallization agent. Then later on, Frankel Conrad also confirmed that the genetic material of DNA is RNA. It is particularly suggested by Frankel Conrad. Then we talk about the classification of viruses. So let's talk about the classification of viruses. When we talk about classification of viruses, one community is there whose name is the ICTD. That is International Committee Committee on Taxonomy of Viruses. What is the name of this particular ICTV? International Committee on Taxonomy of Viruses. And the work of this ICTV is to regulate the norms of classification and nomenclature of viruses. So ICTV is International Committee on Taxonomy of Viruses. Then the ICTV scheme has only three hierarchical levels: the family, including subfamily, and genus and species. Means they are just talk about this three. Means that viruses, if we want to classify, we can get the family, genus, and species. The higher order organization categories are not found here. Then the viruses are named after the disease they cause. So based on this, naming is done. Example, poliovirus, because it causes poliomyelitis. Okay, so this look like this. The family names actually end with viridae. Actually end with the viridae. And the another category is genus, and genus name end with virus, whereas the species name described in nature. So when we talk about isotopy hierarchy, it is known that they have a family, below family they have a genus, below genus they have a species, and these three categories are found. So in this, we know that AIDS is one of the important virus. It's acquired immunodeficiency syndrome virus, and this AIDS is caused particularly by AIDS particularly due to the HIV virus. So if we talk about classification, the family where they belong, that is a retroviridae. So viridae is used as a suffix for the family. Okay. Then the genus is lentivirus. So virus term is used suffix for the genus. And species, that is human immunodeficiency virus. So HIV is the particular species. When we talk about after classification of AIDS virus, we talk about structure of virus, and the structure of virus is the range of size, that is 300 nanometer PMV, 20 nanometer as parvovirus. Okay. So this is the sizes, but parvovirus is not necessary. Not to remember, maybe you remember the particular TMV, that is 300 nanometer size is there. And if you talk about size of TMV virus detail, there if you write actual size, that is 300 micrometer by 20 nanometer, 20 micrometer, or you can write 300 nanometer by 20 nanometer. Such thick is found in case of this virus. Then there are other viruses like helical viruses. Helical viruses resemble long roads that may be rigid or flexible. Then rigid viruses, particularly the example is rabies virus. Flexible virus is tape, particularly helical virus have a helix. You can see this is the diagram of rabies virus, and this is the diagram of TMB. Now, when we see the diagram of this TMB, we come to know that this is tobacomyces virus, and this tobacomyces virus, this is the stranded RNA, and this are the capsomeres. So capsomeres are found to be covering this RNA in between. Then another one is the polylateral virus. Polylateral virus, that many animal, plant, and vegetable viruses are polylateral, means they are many sided. We can see the examples like herpes virus. You can see herpes virus has particular polylateral things. Then the particular polio virus. So they have many sides. Because of this, you can see here, you can see the Okay, different sides are shown here. So this shows the different sides. Okay, so these viruses, so they are polyhedral. Some viruses have an envelope outside their capsule. Usually what happens, there is a centrally located DNA or RNA that is nucleic acid. The surrounded protein capsule. Then surrounded mitosis of virus by the envelope. That type of viruses are known as envelope viruses. When we talk about envelope viruses, these viruses are roughly spherical. They are usually spherical. And the example is influenza virus. You can see the outermost covering is nothing but envelope. Now it is known that this envelope is made up of these viruses. Envelope is made up of carbohydrate, protein, and lipids. Now, proteins are provided by a virus because they have a DNA or RNA that will enter the host, and by using the host machinery, synthesize proteins by their own. So that is why virus. But carbohydrate and lipid are from host. And by accumulation of all this, there is a formulation of envelope. So this virus are roughly spherical and they have a envelope. The example is influenza virus. Then, if we see further, the another group of virus that is known as bacteriophage. And we talk about bacteriophage. Bacteriophages are nothing but viruses that particularly cause infection with the bacteria. They infect bacteria. So the viruses which infect bacteria are known as bacterioviruses. And this bacterioviruses have a complicated structure. So they are also called complex viruses. Apart from this bacteriophage is having a polyhedral symmetry in their head. Polyhedral is many sided. Now helical symmetry in the tail sheath is also observed. Now after this, when we see the tail has helical symmetry, where the head has a polyhedral symmetry. The envelope contains glycoprotein projection known as spikes. Spikes are not usual. Spikes are useful for attaching the virus to susceptible host cell. Okay, so the example is measles virus and bacteriophage. You can see this the diagrammatic structure of bacteriophage. In this bacteriophage, if we locate the structure, you can see this is the head part, and head part has many sides, and within this many sides there is a presence of DNA. So this head is polyhedral. Apart from this, there is a presence of tail sheath, which covers the whole tail. The tail is internally fused. Internally, tail is helical. These are the spikes which are known as tail fibers. And apart from this is the end disc, and here you will find tail fins, which create a pore by landing on bacteria, and you through this pore the DNA of this particular head region of virus enters into the bacteria. So all viruses consist of, is discussed, whatever the virus we have discussed, we know that all viruses consist of capsid and core. Core has DNA or RNA and capsid is a protein compound. When we see the core, the nucleic acid will form genome that present in the central core. Apart from this, we will study that capsid is present, which is surrounding the coat of protein, and this capsid gives the shape and protect the genome. The capsid is made up of protein subunit called a capsid. So you can see if we talk about the bacteriophage structure like this, here Subunit of these capsules are known as capsules. So here one is the capsule and that you will find out nucleic acid. Then the viral genome contains double-stranded DNA or single-stranded DNA. Virus that infect plant generally single-stranded RNA because plants show no specific. Then viruses that infect animals have double-stranded DNA, usually animal viruses. Bacteriophages and other double-stranded DNA. Now, if we talk about these viruses that infect particular plant cells, are known as phytopathogenic. And viruses that infect animals are known as zoophaginic. And the bacteriophages are bacterial viruses that infect they have double-stranded DNA. Usually viruses have a double-stranded DNA as well as, but the plant virus have single-stranded DNA. Usually. That viral nucleic acid molecules are either circular or DNA can be either circular or linear. Most of the viruses have a single nucleic acid molecule, but if you have more than one nucleic acid as well, suppose we take the example of HIV. HIV has two identical molecules of RNA representing its genomic copies. Means in HIV virus there is a presence of two copies of RNA. And obviously that is RNA virus that is a retrovirus. So you can see this is HIV virus and this papilloma virus, which are so observed like this. So students, what we have studied till now that we should understand that in central region there is a presence of what nucleic acid, that this is covered by a layer of capsomere and that will form what capsid, and this surrounding to this will find a presence of what that we have discussed now. So it is nucleic acid. This is capsomeres. And this capsomeres form capsid. And this is the layer. Okay. So this is the particular structural part that we discuss. Now we will talking about the detailed structure of some viruses like TMB. And you know that TMB is a topicomosic virus. This topicomosic virus, if you see, is about 300 nanometer long, 18 nanometer, that is 300 by 20 nanometer, also be right sometimes. Its molecular weight is 39 to 10 to 6 dalton. Capsid is made up of 2130 capsomeres. Each one that is one of the important points that is mentioned. How many? 2130 capsomeres are involved in the capsid of this particular. But in TMB, if we see the capsomeres, as you can see this is the diagram of TMB. As we have discussed, in TMB there is a single-stranded RNA and surrounded by a protein capsid. This each unit is known as capsomeres. Now, the capsomeres are identical in size, arranged in helical manner, and around the central hollow space of 4 nanometer. Each capsomere is made up of single polypeptide chain with 158 amino acids. How many? 158 amino acids. Now, inside the protein capsule, there is single stranded virally coiled RNA molecule which consists of 1600, 6500, you can say nucleotides. And how many capsule pair was that? 2100. Now, if you see in this diagram or in the structural part of TMB, then it is known that in TMB there is 6400 to 6500 nucleotides and 2130 capsule. So the ratio is something 3 is to 1 between, you can say, nucleotides and capsules. So this is the thing. Then, if you talk, in TMP it is known that they have 5% RNA and 95% protein. That is present in particular protein. Then there is another one is the bacteriophase. Bacteriophase, if you talk about, there are two bacteriophase. And in bacteriophase can be distributed into head and tail region joined by polar. Tail includes gills, tail sheath, base plate, and tail feathers. And this is the structure. And in this structure, we have a head, muscle, and there is a neck, there is a collar, we have a tail region that is covered by sheath, there are tail fibers, there is a base plate, there is DNA, and there is tail fins. So this is the structure of probacteria in which head is the capsule part. Head is the capsule part which is polyhedral shape and ciliate are capsule layers. So T4 bacteria. Then the tail helps virus attach to through the tail or tail fibers. The virus first attached to the host cell. Then the tail sheath add injecting viral DNA into the host cell. So tail sheath help to push the DNA within the. So like this, the bacterial cell wall is there, and over which the bacteriophages are seated. They will create a pore, and through pore, the particularly DNA is entered the host. You can say like this process. This is the bacteriophage is pushing the DNA into the host cell. Now we talk about some of the examples who crystallize DNA. So its answer is the particularly Stanley, W. M. Stanley, who was a scientist who specifically crystallized the steel and for which he awarded the Nobel Prize. So the answer becomes W. M. Stanley. Then another some questions like we consider the virus resemble long road R. They are particularly helical viruses, as helical viruses resemble long roads which may be rigid or flexible. The third question is what are the example of envelope virus? Envelope virus are the herpes virus and the HIV virus. So answer becomes herpes virus as well as in influenza also this is shown. So influenza is a more appropriate, but it's a herpes symptom. It is a herpes virus that has the envelope. HIV has the envelope and influenza virus. So influenza virus become correct. The viruses which infect bacteria are the viruses which are formed of two factor bacteria are known as bacteriophages. Then The another question is spikes of bacteriophage are useful for the spikes of bacteriophage are useful for the attachment to the host cell. So if this is the answer, the spikes are useful for attaching virus to the host cell. So till this is some of the particular part of the viruses. Now after this, we are going to discuss the particular life cycle or reproduction strategy in case of viruses.